to wait for every day rather than the final panel. Maybe you have this place where you're allowed to be bad. Maybe, I don't know. Isn't that the time of night? Never done it here. I don't think so. My son, who is allergic to tree nuts and shrimp, says, or seafood, says, heaven's going to be pecan crusted shrimp. So that's going to be his eat and can't eat. All right. So we are on a quest to understand histology. So today will be, I believe, going to be the finish of histology. Then we are going to take a couple tissues and we are going to make our first organ. And that first organ on Friday is going to be our skin. Okay? If you were not aware, skin is an organ. We're going to put that couple tissues together and start talking about skin. And so after that, after this next exam, everything we do is going to be organs and organ systems. So we're going to finish our foundational material after today. That's the hope. So currently, if you are not playing along, genetics and rheology, histology, you ought to be familiar with those. You ought to be studying those. You ought to be ready and able to take an exam over those. Today, if I were to say, let's take an exam today. Be able to do those three. Okay? Not, then you still got a week to go. All right? Well, actually a little bit more. Because recall, this is important. Right? Attention. There's a few people who were not here the other day. And there's a schedule change. Okay? You with me? Ready? And that schedule change is that there is no class on Wednesday, October 4th. Because as we discussed on Monday, you are all, for the most part, required to be there. Okay, which is Raven Edge Career Fair. And you're saying, but I'm a freshman. It is never too early to start. And for many of you as sophomores and freshmen, you could find summer employment. Whoa, that would be great. Or internships. Wow, that would be cool. And so a lot of employers will be there that will offer and have opportunities for those. Or you could start whittling it down. Do I want to work for St. Francis or St. Vincent, or do I want to work for community when I graduate? Let's start that process. Okay? So that means there is no class on Wednesday. No physical class. Now lab, I'm still trying to figure that out. That means that exam number 2 is moved to next Friday, a week from Friday instead of a week from today. Okay? I have not changed that on the homepage, and I need to do that. But yes, let's. Okay, any questions about that? Why are you touching what? There's always a group in class that just can't pick up the giggle. It tends to be over here. All right. That's all right. Maybe we need to get okay. And then there's one right in the center. All right. So, any questions about that? Any other housekeeping we need to straighten out, deal with? So, by the conclusion of today, we will have finished talking about connective tissue, both what is called loose and dense. And for many of you, these are going to have a little bit more relevance than just talking about epithelial tissue. Trust me on that. We're also going to talk about muscle tissues, which, again, have a lot of relevance to most of you and what you're doing. And we're going to talk about some cell junctions that are found in tissues. And then finally finish with the process of tissue growth, how the tissues grow. There are several ways that they go about growing, so we'll look into that. So as I said, by the time we are finished today, if my estimate is correct, we will have finished tissues and be ready for skin on Friday. So, if you recall from our discussion on Monday, or for those of you who are not with us, but to bring us up to speed, histology is the study of tissues. And to make life very easy, there are only 4: epithelial, connective, nervous, and muscle. And when I say that there are only 4, I mean 4 types, 4 categories. Because as we saw yesterday or on Monday, there is a plethora of epithelial tissues. There's not just a epithelial tissue, there are many. And the same is going to be true for connective, a little bit for muscle. Nervous is pretty straightforward, as we'll see later on. So that's our definition. And if you like comparisons, use this table. It's in your PowerPoint that is online. So you can certainly refer back to that for organization purposes. And that's how our tissues began. Remember, there was a connection between starting with embryology and the study of tissues. That at one point, roughly 2 weeks, you were 3 layers. 3 layers. You were flat disc, 3 layers. Upper layer, ectoderm. Middle layer, mesoderm. Bottom layer, or deepest layer, endoderm. And our tissues came from that. Okay, the tissues we see in the adult organism came from one of those 3 layers. When we describe the epithelial tissues, we are primarily talking about things from the ectoderm and the endoderm. Those are our 2 sources. Today, with connective tissue, we're going to spend quite a bit of time talking about tissues that came from the mesoderm. Okay, because they're middle. I mean, if you just think about, bones, where are those? Well, we're kind of in the middle. Right? They're in the middle. And that's where that tissue came from. Mesoderm, the middle. Okay? That's a simplified approach, but it's fairly accurate overall. All right, so we also said that tissues are composed of these things, regardless of which tissue we talk about. They are always going to be cells that are present, and a matrix, background or area, the material around the cells, and the cells of the tissue, for the most part, are producing the fibers that we see in the tissue. So in this particular tissue, which is the cartilage, we see the little black fine lines. Those are the fibers. Those are proteins that have been stained. Those were made by the cells that you see in this tissue. Okay? The exception to that rule is blood. Most of the fibers in blood, most of the proteins in blood were not made by the cells of blood. Made by the liver. And then there's the ground substance. This is the fluid component that you see around the cells that are where the proteins are found. And in that, there's water, minerals, gases, nutrients, and waste. Now, in large part, how we categorize these tissues is not just solely on their source, on their origins of endoderm, ectoderm, mesoderm, but also the ratio of how many cells to matrix. Some of the tissues, as we saw the other day, epithelial tissues, lots of cells, very little matrix. Okay, it's just very little, which is a good thing, because we want an epithelial tissue that is solid, or that is continuous with cells, because of its protective nature and other roles that it does. Today, we're going to kind of flip that on its end, okay, because this is a connective tissue. There's not a whole lot of tissue or cells in that tissue, all right, but a lot of matrix. So we're going to look at how that changes. So as we defined, epithelial tissues are always on the surface. Name any surface, and I'll tell you what epithelial tissue is there. It's not a case. But every surface has an epithelial tissue. And epithelial tissues are usually on loose connective tissue. We don't know what that is yet. We're going to look at it. And there's usually something called a basement membrane. It's what anchors the cells of the epithelial tissue. And as we were talking about with our paper cuts and our hot pizza cheese things that happen to us, we don't bleed if all we cut is the epithelial tissue. Why? Because blood vessels. They are non-vascularized, which means that the way they obtain their nutrients is diffusion from the loose connective tissue, which they are resting on. So diffusion is the process by which they're going to be nourished. And we said that they are innervated. Paper cuts hurt like heck, especially if you get lemon juice in them, right? Hurts. Hot pizza cheese, oh God. Burns, painful. Okay, but no bleeding, because there is nerve endings. All right. So, connective tissues. Those are the functions of our connective tissue. And over the next few minutes, as we go through our different connective tissues, try to assign a function to that tissue, basically from that list. Because what I want you to know about each of the tissues, each of the specific tissues, where is it, and what does it do? And can you generally describe it? Now, those of you who were in lab yesterday had an opportunity to kind of do that, all right, without my guidance, per se. It was an episode, or an exercise, rather, in for you thinking through how do I answer those three questions. What does it look like? Where is it? And what does it do? So this is an opportunity to assign a function to a tissue. All right, so these are some examples. We're going to see more of those today. Broad overview of where connective tissues come from, as we said, from mesenchyme, which is a derivative of mesoderm. And we see that we're going to do connective tissue proper, cartilage, bone, and blood. Those are the targets, those are the things we're going to take a look at today. All right, so connective tissue proper. In other words, this looks like connective tissue. When they set out to say, what is connective tissue? This was kind of their base definition. So that's why we call it proper. And it's the tissue that really genuinely connects things in a connected sort of manner. There's loose, which is areolar, reticular, and adipose. Okay? Those are our three types of loose connective tissue. Now, when we say loose, we immediately say right below it, there is lots of ground substance. In other words, lots of matrix and not as many cells. That's the definition of our loose connective tissue. And as we've always said, if I'm defining something as one way, then there is likely the opposite or another way. So the opposite of loose is going to be dense. So loose connective tissue is areolar. And as I try to emphasize when I see that word or say that word, airy. Lots of space. It's airy. It's open. And when we see that it is very vascular and underlies the epithelium, lots of blood vessels. Paper cuts don't bleed, but if I go a little deeper, sure they bleed. Right? Because now I get that loose connective tissue where there is plenty of blood vessels. Reticular tissue is going to form a framework, a scaffolding for particular organs like lymph nodes, thymus, spleen, bone marrow. These are all tissues of the immune system or lymphatic system. And we have blood and fluid that goes through there, slowly moving through because of the spaces that are found there. And then lastly, adipose tissue, that's your fat storage. There are two varieties of that. We're going to talk more about it in a second. So that's the broad overview of loose. And like we did the other day, we're going to see pictures and talk about locations of each in a second. Okay. Are we ready? That was our summary from last time. Okay. So that is the artist's rendition of loose connective tissue. Now, this loose connective tissue in this particular drawing has every cell type that you would ever expect to see in loose connective tissue. And probably what is not shown is that your skin, your epidermis, would be up here. So the tissue that is part of your skin, the epidermis, and epithelial tissue would be right up here. Lots of blood vessels in this tissue to supply you with blood. But there's also going to be a lot of cells that look kind of weird, like these neutrophils here, and a lymphocyte, and a macrophage. These are cells of your immune system. So that when you get through the epidermis, this continuous covering of your skin, this epithelial tissue that covers you and protects you, you break through that, you have the cells of your immune system to protect you. And they're right there in the loose connective tissue. So that's the process by which you're going to see inflammation. Okay, that's where inflammation is going to start. That's why those cells tend to be there. You also see that there are nerve fibers. Remember we say that epithelial tissue is innervated. Well, we have to have that nerve fiber get there, and it travels through the leaves. But again, we see lots of blood vessels here, and most of the fibers are one particular kind called collagen. So, there's our loose connective tissue, the actual micrograph of it. So this is a picture that I can show you from any of the tissues I have upstairs on the slides. And you see again that they have stained it. These bright red ones are collagen, and these little thin ones that are black are either reticular or elastic. Because, hey, guess what? I pull on my skin and it snaps back. That's great. That's good. Okay. Now, if you were to buy some of those expensive creams that contain collagen, collagen cannot get into this tissue. It can't be absorbed through your epidermis. Don't buy it. Don't bother. Okay? Money wasted. Same. Okay? Just avoid those makeup counters and just walk on through and say, "No, I'll drink water. Thank you very much." All it over. Okay? Now, where do we find it? Well, pretty much everywhere because it's underneath the epithelia. So, hey, where's epithelia? Oh, everywhere. So if it's underneath the epithelia, then this tissue is everywhere as well. So it's probably the most common connective tissue we find. So if I were to ask you where, you pretty much can say anywhere. There's adipose tissue. Now this is white adipose tissue. This is the storage of lipids. This is where we store extra energy. Okay? So as you eat and consume your Chick-fil-A over there, and as you consume that, if you are not burning all those calories off per day, then there's extra glucose and extra lipids that you've consumed. You're not needing them for energy today. Your body says, no, we're not going to put those into the Anderson wastewater system. We're going to store them. You know, there may come a day when we don't get enough energy in our diet. So we store it as lipids. Now, this is showing a breast tissue as one location, but there are many, many, many other places, obviously, and most of it is just deep to the skin. So when we inject something sub-Q or subcutaneous, that's where we're putting it. It is a lipid-soluble injection because we're putting it into the lipids of the adipose tissue. So, you can see it's under the skin, it's around the kidneys and the eyeballs, within the abdomen, in the breast. I mean, wow, lots of locations. Okay. So, adipose, white adipose. Now, let me tell you, let me back up. Sorry. The other variety is brown adipose tissue. Brown adipose tissue. It looks a little different, and I'm not going to worry about the differences in terms of the look. You all still have a little brown adipose tissue, but you primarily had more when you were a newborn. Because brown adipose tissue does not store lipids the same way, but rather it converts the lipids into heat, directly into heat. And that's how a newborn generates some of its body heat. Realize you're coming out of a nice, warm, and 37 degrees environment into 25, 28 degree room. Okay? And it's cold. And so this is there to generate heat. Okay? Not energy, but heat. So over time, you have needed less brown, and so it has gone away. A study done about 20 years ago in Scandinavia, they turned down the house temperatures to about 55 to 60 degrees and told them, wear shorts, wear short sleeves, don't bundle up. Don't do anything different, just do that. And people lost weight. Not a lot, they did lose some. In an effort to generate heat during those months when they were in that situation. I don't know. Keep trying to say that's what we ought to do at the house to save money as well. She doesn't buy it. She'll just get out her space heater. Yeah. Correct. Right away. They lose a little bit of their weight. That's part of it, certainly. All right. Reticular tissue. This is your spleen. Spleen located upper left quadrant, right over here on the lateral side. And you can see that there is a blood vessel in and a blood vessel out. So there is a lot of blood going through this particular organ. And part of the function of the spleen, as we'll talk later on, is to break down worn out old erythrocytes. Because your erythrocytes, your red blood cells, last about 180 days. So you need to get rid of them. And the spleen is one of those organs that does that. So here are the reticular fibers, these black fibers. That's forming that framework. And then all of this are either red blood cells or leukocytes, that is white blood cells, percolating, moving through there, being monitored, and we are removing the worn out red blood cells, among other things. Okay? So, lymph nodes, spleen, tonsils, those are where we find reticular tissue. All right, so if there's loose, then there's dense. So the big difference, instead of a lot of matrix, there's a lot of fibers. Lots and lots and lots and lots and lots of fibers. There's dense regular, and there is dense irregular. Whoop, sorry. So, how many of you have torn a tendon or a ligament? Yeah. Sorry. Okay. Now, how many of you had to have that torn tendon or ligament replaced with another tissue somewhere else in the body? Anybody get a cadaver? Yeah, I have a student who had a cadaver ligament inserted into it. Okay? Because they couldn't find the adequate what they needed. Okay? There's a reason why. We'll explore that in a second. So tendons and ligaments, collagen fibers run parallel to each other. They're all in arrangement, nice and neat bundles. Nice and neat bundles, parallel to each other. Very organized. And you guys kind of already know that because you eat chicken. And you get a drumstick and there's some tendons. And they're nice and straight, cord-like in some cases. Okay? That's tendon. Or at the ligament end, at the end of the bones where they're connecting. Right? Drumstick to thigh. That's the ligaments. So you're aware of these things. What's the big idea? They have few blood vessels. Why is that so important? Did your ligament heal on its own? Quickly? No. Jackson, they had to replace it. Why?
Because there's no blood supply.
And it wasn't going to heal on its own. Very quickly. Right? Break a bone, 6 to 8 weeks. Tear a ligament? Yeah, months. Months. Okay? It's not a quick healing process because there are few blood vessels. Few blood vessels. Right? Aaron Rodgers tore his Achilles tendon. Do you think he's going to come back in time in January and make it to the Super Bowl? No, because Zach Wilson's the quarterback. But there's no way he would be ready in January or February. It's just not going to happen. Especially at 39 years of age. If you're an Aaron Rodgers fan, I'm sorry, but that's true. All right, so this is what one looks like in terms of dense regular connective tissue. Look at that. All in bundles, all parallel to each other, and very, very limited amount of background material. See, I'm stuck to that. All right. So when you tear this, that's why it's not going to replace it. It just isn't going to be the healing properties of this tissue. It's not present to help it heal. Now, you'll notice that in this preparation on the micrograph, it's got some waves to it because this tissue does stretch. Okay, there's a certain amount of stretch. There's a certain amount of twist. But don't plant your foot and turn and not have the foot go with it, because then things tear. Right? That's what happens. All right. Then the dense, irregular tissue. So instead of the bundles running parallel, there is not really a set arrangement to them. They're pretty much all over the place. And it forms what we call the dermis. That's deep to the epidermis of your skin. We'll look at that on Friday. It forms capsules around your organs. Why is the kidney the shape of the kidney? Because of the capsule, because of this dense, irregular tissue. And so it will look like this in a second, but also, again, few blood vessels. So it looks like that. Right? All over the place. Not necessarily any set arrangement. Okay? So this will withstand some twisting. This will be allowed to expand, to distend, to get bigger in some way, stretch a little bit, okay? Without tearing. All right, so those are our dents. Now on to cartilage. Anybody torn any cartilage? You and I both. Okay. Left knee, yeah, I don't know how much is left. So cartilage is a tissue of support. It's a tissue that is going to be involved in our locomotion. And it's a tissue that binds things together, as we're going to see in a minute. And there are three varieties of cartilage. There is what we call hyaline cartilage, the most common of the three cartilages. It contains collagen fibers, again. And in this case, some people will try and eat cartilage. They'll get capsules of cartilage to eat and think that might help their joints. I don't know too much about that, whether that's true or not. Okay? Because collagen's a protein. And if I eat a protein, I'm pretty much thinking I'm going to digest that protein. So I don't know. Okay? But I could get cartilage injected into my joints. That could work and has been shown. So, or collagen injected. It's found as an articular cartilage, a tracheal cartilage, the larynx, and the costal. Well, trachea, windpipe, larynx, vocal, costal, connect ribs to sternum. Ribs to sternum. Articular, end of the bones. All your long bones have a covering of articular cartilage that is hyaline. And that's what's missing from my left knee on my tibia. Okay? One out, one gone, almost gone. So that's what's helping you stabilize, cushion, and move. Now, hyaline cartilage looks like this. I'm going to just pop that up. And you can see that there's a lot of matrix in there. Some cells with a lot of matrix. And the cool thing about hyaline cartilage, like in the knee is a great example. You push down on it when you're standing, when you're walking, and the tissue gets thinner because it squishes. That's a technical scientific term. It squishes and the water goes out. Okay? Then when you release that tension, like sponge, it absorbs the water. Now, I'm not talking like volume, like golly, liters or anything, just a little bit. And that's what keeps this tissue, that movement of fluid in and out, being a little more supple, being more able to lubricate the joints and keep them moving well. Okay. Now, costal cartilage between the rib and the sternum don't really experience that as much, and that costal cartilage can get brittle over time, more so quickly than other tissues, because we're just not using it. So, hyaline cartilage, all over the place, as we mentioned. Whoops. The next one is elastic cartilage. Elastic cartilage. And it contains this kind of, wow, really? Elastic fibers. Guess what it does? Yeah, really? Okay. It has some elastic properties. One of the best places to find it, because there's not a lot of it, is your external ear. So when you go walking through the woods, the branches try to grab your hair and your head and ear, if it doesn't grab them, looks great for your sons. Maybe I get that. I don't know. Or your sisters, right? Or your brother. Bing! All right? Now, how many of you don't have to show your hands, but how many of you have got your cartilage? Pierced. Right? Up here. Right? It's never going to heal. It's going to be there forever. Okay. Better like it. Because when you take whatever out, it's just not going to heal. Sorry. It's going to be there. All right? Because what's not present? Blood vessels, right? It just isn't going to heal very well. Okay? So think twice if you're piercing your cartilages. Okay? Some of you know that your earlobes may have closed up. I can still get an earring in there if I wanted to. I haven't done that for a while. Maybe I'll come into class one day. I could. Yeah, go find one. What's that? Please don't. Oh, do? Yeah, I'll take it. All right. And then fibrocartilage. I'm going to show you pictures of these two in a second. Parallel collagen bundles. Well, that sounds like our dense regular tissue. It is very similar, but it is enough different. And we find it in only 3 small little locations. It's in your pubic symphysis, between your pubic bones. So it's what holds your pelvis together. That's probably a good thing. Okay? Very strong tissue, does not twist, does not tear very easily. It's also found in your menisci. Meniscus, a little ring of fibrocartilage on your knee, for instance. It's also on your TMJ joint. So it's on your temporal bone, forming a place to hold your mandible. And then intervertebral discs, discs between your vertebrae, also has fibrocartilage in it. Okay? Strong tissue, doesn't tear very easily. So this is your elastic cartilage. Those are the elastic fibers, these black fibers. Okay? A few more cells than in hyaline. And then this is the fibrocartilage. And except for the cells in there, more cells than what we saw in a tendon, that's pretty much like tendons. So they're very similar. All right. So those are our cartilages. Hyaline, elastic, fibro. Hyaline most common. Okay, the one that we're probably going to interact with more than any of the others in our lifetime. Okay, especially if you're going into the medical field because of articular cartilage. Wearing away and creating osteoarthritis like in my knee. Or the other one, elastic in the ear and fibrocartilage in our pubic symphysis and intervertebral discs. All right, so those are the big ones. There's others, but these we're going to kind of cover separate, on their own, not go into too much detail today. We're going to have a whole discussion about bone. All about bone, just bone, and nothing but bone. Okay? So you just need to know for now that there are two, the bone kind of looks 2 different ways. It's either compact or spongy. That's all I'm expecting for right now. The other one is blood. Yeah. That's the first unit after Christmas. And we'll spend the whole week talking or more talking about blood. So it is a connective tissue. We'll deal with that on its own. So there's a picture of bone. There's a picture of blood. Got it? Then we have what are called the excitable tissues. And so excitable tissues respond to stimuli. They respond to stimuli. So they are able to react. There is going to be something about this tissue that is going to change. And those are the two. nervous tissue and muscle. And guess what? We're going to look at those on their own. So we're going to have a few units about nervous tissue on its own. We're going to have a whole unit on skeletal muscle. And we'll have a whole unit on cardiac muscle. So again, we're going to talk about those down the road. Now, please note one important thing under muscle. How many of you have ever heard striated muscle? Yeah, they're all striated. Every last one of them has striations in it. So don't use that term, please. It's skeletal. Where's that? Well, the skeletal muscles. Cardiac muscle, where do we find it? Smooth muscle. Who said it? Where? Internal organs, yeah. There are lots of other places, but yeah. Digestive, respiratory, urogenital, okay, all those places. All right, so there's a picture of nervous tissue, and as I mentioned, we're going to get into that in detail about a month or so. So these are neurons, and those are axons, the little black things. And then that's skeletal muscle. Gosh, that looks striated for sure. Okay. Oh, wait, there's cardiac muscle. I see striations. Oh, look at that. And then, oh, that's smooth muscle. And you got to look real close, but guess what's in there? Striations. Okay. It just doesn't appear as striated as the first two, which is why we label it smooth. Okay. So again, that's kind of all you need to know. How many muscle types? 3. What are they? Skeletal, cardiac, smooth. All right. Now, sometimes we use these terms. Cutaneous and mucus. Or mucosa. Where are those? Are they anything different? No, they're not anything different. It is, where is the epithelial tissue located? That's really all this is saying. So when we talk about cutaneous membranes, we're talking about the epidermis of the skin, that outer layer of your skin. That epithelial tissue on your skin. And it's dry, right? Oh yeah, it's dry. Okay? There's no fluid there. And it's waterproof. Great. Cool. The other one, mucosa, is, gosh, in contact with mucus. And so we find that like in the respiratory system. And we find that in the digestive system. And we're just calling those mucosal membranes. That's it. The epithelium that is in contact with mucus. Just the designation. Nothing more. But in your clinical work down the road, you may hear and talk about those things. So then we described, and these are back to, like a month ago, serous membranes that line our cavities and line our organs. And serous is saying that it's in contact with the fluid, or the watery fluid. Right? So when we think about the peritoneum, then that is in contact with the fluid of your abdominal cavity. Or it's parietal fluid. It's in the portal. All right. So just again, some designations. Now, a tissue is made of the same type of cells as we've kind of looked at, seen over the last 2 days. But there has to be a way to hold those cells together to create a tissue, especially an epithelial tissue. And we don't want epithelial tissues to have the cells separate one from another. We want them to lock together. We have what are called junctions to hold them. And those junctions are made by the 2 adjoining cells. So the 2 neighboring cells are going to help form these junctions. And there are 3 of them. First is called a desmosome. And for kind of a quick, easy way to reference it, think about it, it's kind of like a spot weld. In other words, you take 2 membranes, like you see here, and you fuse those 2 membranes together, right there in that one little spot. You weld them together. How are they welded? Because both cells produce proteins that interlock at an angle.
desmosome patch. And I also note that it does not encircle the cell, which means, oh, there's probably one that goes all around the cell. And that's called a tight junction. Sometimes referred to as a belt. And it encircles the cell like these. So if we look at our cells here, we see this going all the way around. Imagine that if I pick that cell right there, it's in contact with 1, 2, 3, 4, 5, 6 cells. All the way around it. It would be as if I took my belt off and took 6 people and looped it through all their belt loops and my belt loops, and we walked together. We're all linked together. Okay? Now the big, huge idea is it seals off what we call the intercellular space. Because now, these 2 cells that are joining are sealed off by the space in between doesn't allow for passage of things. Why is that important? It means that anything that's going to go from this surface to this surface has to go through the cell. It's going to regulate what enters the body. So we find tight junctions in the epithelium lining your digestive system. It seals it off. So if you want to only absorb glucose, that's all you absorb. You don't mess around with the bacteria that you had in your Chick-fil-A. All right, so those are 2. Means there's a third. And the third is called a gap junction. Gap junction. And what that is a communicating junction. So here's my little gap junction right down here. And literally, it is a pore between the cells. It is a pore between the cells. And that pore can open or close, depending upon conditions. And because we're just so good at thinking up imaginative names, the proteins are called connexons. What do? They call them connexons. How about that? All right, so it is connecting these cells, but what is it doing? It allows ions to move from cell A to cell B. They go through the pore. Why do I want that? Because they're found in places like cardiac and smooth muscle, these excitable tissues. So when we start talking in a month about action potentials, which is the movement of ions, they're going to play a major role in communication between the cells. As you know, your heart all contracts at the same time. Why? Gap junction. Yeah, that's where we're kind of headed. Very important. Okay? Almost. A little bit more. So there's the overview again of the 3. Desmosomes, tight junctions, gap junctions. And if your desmosomes don't work because of a genetic condition, you might have something called pemphigus vulgaris. So the epithelial cells of the skin are not welded together and they're not held together by those desmosomes. And fluid under conditions, certain conditions, accumulates underneath and pops it up like that. And you can see that they eventually might pop and crust over. Okay. Steroids are a general way to cure that, or not cure, but treat that. It's not necessarily life-threatening unless you had a huge amount that opened all at once. So, epithelial tissues create and form glands. Glands secrete things. They produce and they secrete. Endocrine glands are one type of glands, and we're not going to talk about them today. We'll talk about them when we get to the endocrine system. For now, all you need to know is they secrete their products into blood. That's it. That's all you have to know.
The second one you do need to know a little bit more about. Those are exocrine. Exo, outside. Crine means secrete. So these secrete outside. In other words, they secrete onto an epithelial tissue. Sweat gland, mammary gland, pancreas. Those are all exocrine glands secreting onto a surface. Now they can be simple or they can be compound. This one is compound. Multiple branches. They can be tubular, just a straight tube, or what we call acinar. This is an acinar. So that means that there are little endings, and that's where the production is taking place. And then, more importantly, how do they secrete? What is their process of producing a secretion? If it is a merocrine gland, it is a secretion of filtration. Sweat glands are a merocrine gland. You filter your plasma to create sweat. Holocrine, on the other hand, is a portion of the cell or the entire cell. Mammary glands are holocrine. They produce a portion of their cell as the secretion. And so this is the simplest of all. That's the goblet cell, one cell. That's the designation. You can see simple. You see simple branched. That's alveolar or acinar. And I'm not going to ask you all these, just show it. Right? I don't even ask my histology students. And that's the difference in secretion. Filtration, right here. Vesicles that release, or portion of the cell. All right. We will pick up almost made it on Friday with this. Emily's leaving.